last day we practiced dihybrid cross. Okay, normal cross we have the ideas, is it or not? Now going to some tricks, tricky part. One tricky part I have discussed regarding the gamete formation. How to form the gamete. Now the second tricky part, without if. Understand that tricks only will be applicable if the situation is a Mendelian cross. If situation is not the Mendelian cross, then these tricks will not be applicable there. It's not given in the book. As much as you will practice, maybe you can also discover some types of tricks. These all are the tricky part from my sides. But it will be only applicable if that is a Mendelian situation. Otherwise, it will be not applicable. Now tell me, monohybrid cross. Monohybrid cross, standard Mendelian cross. What is the phenotype ratio? What is the phenotype ratio? 3:1. In standard Mendelian cross, what is the genotype ratio? Yes, genotype ratio will be 1:2:1. Very good. Chalo, that we have to only remember. That only we have to remember, understand that. Suppose dihybrid cross, one time came, dihybrid cross ratio ask and you forget in the class, in the exam hall. Situation if normal, then and there you can calculate without the calculation of dihybrid cross. How? 3:1 into 3:1. 3 3s are 9 is to 3 1s are 3, 1 3s are 3, 1 1s are 1, 9:3:3:1. Chalo sir, I can easily remember 9:3:3:1. I don't need these tricks. Chalo, very good. What about the genotype ratio? Genotype ratio, how you will able to remember? So same process, 1:2:1 multiplied by 1:2:1. So I will multiply 1 with the entire. 1 with the entire. So 1:2:1 is 2. Then I will multiply this 2 with this entire. So 2:4:2 is 2. Again I will multiply this 1 with the entire. 1:2:1. Done. Here I have not solved any problem. Situation if Mendelian cross. Understand that Mendel was not biologist on the day when I discussed that part. Whatever Mendel did, it's based on statistical mathematical formula. So everything is based on mathematics here. Clear? Understand? If the situation is normal, then these tricks will be applicable. Otherwise, it will not be applicable. Okay? Now, this is first part. How we can get the ratio. Some question might be asked, like how many offspring produce in monohybrid cross? Total number of offspring in monohybrid cross? 4. 4. Is it not 2/2? So obviously 4 offspring maximum can produce. Now question can be asked, let's say if this is the box checkerboard, if this is the checkerboard, okay, these are heading purpose, forget about that. So how many box? 4 box. So 4 offspring. Now every offspring probability is 25%. Remember that. Every offspring probability is 25%. Every offspring probability. So that means total is 100%. So how I have calculated 100 by 4? Total number of member is 4. 100 by 4 means every box probability is 25%. Like Mendelian monohybrid cross F2 generation ratio 3:1. So what is the meaning of this 3:1? This 3:1, 3:1, 3 means 25 into 3, 75% is to 25%. Huh? So if it asks in ratio, I think it's very easy. Just in a simple, I am not going to the base of the mathematics. Just understand 4 box is equal to 100%. Each box probability is 25%. Bas. 3 tall, 3 into 25, 75%. 1 dwarf, 25%. Clear or not? Huh? Clear? Okay. Chalo. Another part. Be careful about one dihybrid cross. Just tell me which dihybrid cross we have done in the class. Tell me. Which one? Tall and purple. Na? So, if the situation now, be very careful and try to understand that. If parental generation given capital T, capital T, capital P cross with small t, small t, small p, small p. So, the given situation is talking about tall and purple versus dwarf and white, dwarf and white, tall and purple versus dwarf and white. Or if the situation is given capital T, small p cross with small t, capital P. So here tall and white cross with dwarf and purple. Understand that? Getting my point or not? Here F1 generation, I am not going to the gamut, I am step jumping here. Here F1 generation will be capital T, capital P, small p. Here also same will come in the F1 generation. Remember that capital T, capital P, small p. Same to same. So both the cross is ultimately same in F1 and F2 generation. It look like might different. This one. NCERT has given this process. Getting my point? That all are dominant versus all are recessive. If we do, then whatever the F1 will come. Here also, this is dominant and recessive. Understand that? Dominant gene, homozygous dominant gene and homozygous recessive gene versus homozygous recessive and homozygous dominant gene. Their outcome is also same. It might be look like different. Their output is always same. Same checkerboard, same each and every single write up will be same. Getting my point or not? Please try to understand whatever I am talking about. Every time we practice this situation, all are dominant versus all are recessive. But this situation also, although it's look like different, but outcome is same. Both F1 generation is same here. Heterozygous tall, heterozygous purple. There is also heterozygous tall, heterozygous purple. So same ratio, same checkerboard, all will be same. Clear? And for any cross, monohybrid cross, dihybrid cross, please remember, if situation is Mendelian, then standard procedure will be applicable. If situation is something else, different process will be applicable. Clear? Okay, write down one question. Standard dihybrid cross. Based on standard dihybrid cross, I am giving you a situational cross. One situation. Upon the raw material, if active C gene work, upon the raw material, if active C gene work, then the color of the flower will be chromogen. color of the flower will be chromogen, that is white. Color of the flower will be chromogen, that is white. Write down question. Upon the raw material, if active C gene work, upon the raw material, if active C gene work, then the color of the flower will be chromogen. Upon the chromogen, if active P gene work, then the color of the flower will be purple. Then the color of the flower will be purple. Upon the raw material, if active C gene work, then the color of the flower will be chromogen, that is white color. Upon the chromogen, if active P gene work, then the color of the flower will be purple. Find out F2 generation phenotype ratio. Find out F2 generation phenotype ratio. Find out F2 generation phenotype ratio. Acha, be sure what is the meaning of active C, active P. First thing, gene active. What is the meaning of gene active? I am tall or dwarf? First thing. Always remember to solve before anything, any question from Mendelian cross. Always remember me. I am tall or dwarf? Tall. Height gene is active within me or not? Yes. So if height gene is active within me, that means either I am capital T, or I am capital T, small t. Getting my point or not? That's why I am tall. That is the meaning of active gene. Active gene means they will give character. My character is tall. That's why either capital T is my genotype or capital T, small t is my genotype. If you remember this line, Sir is tall, you will get all problem solution. So active C gene means either it will be a capital C or capital C, small c. Active P means capital P or capital P, small p. Clear? Understand na? Okay. Chalo. See. So your F1, this is standard cross. No change. Only the change is given, some situation given. Remember that. Only the change which is given, situation given. If you practice clearly in your house dihybrid cross, the way that I discussed, it will come F1 generation. I am just step jumping here. It will come in the F1 capital C, small c, capital P, small p. So F1 cross will be capital C, small c, capital P, small p, cross with capital C, small c, capital P, small p. Now, so in the F2 generation, whatever will be produced, here I am asking about the ratio, phenotype ratio. Understand that. So if you be careful about the chart, checkerboard. Last day I told you, if the chart is normal, standard Mendelian cross, then we will not go to solve the chart because we know about the tricks. What is the tricks? This is the first place where all are capital. Nah? This is the place minimum 1C, minimum 1P is capital. We know these are the 9 locations. So these are the 9 locations where minimum capital C is there. Second position does not matter. Because you know my height is tall. If I get minimum 1 capital T, I will be tall. So second does not matter. So let's say from my father side I have get capital T. It does not matter that from my mother side whatever I will get, I will be ultimately tall. Getting my point or not? So second position does not matter for the dominant character, phenotypic character. Okay. So second position here might be capital, might be small, that does not matter. That's why I am indicating dash. Minimum 1 capital P, second position does not matter. So if C active, P active, which color will come? C active, P active, which color will come? Purple. So how many purple in your checkerboard? 9. Because ultimate Mendelian dihybrid cross ratio is 9:3:3:1. Getting my point? Same ratio will come, but situation will be changed. Because situation I have given here. Understand that I am ultimately asking you what is the phenotypic ratio of F2 generation. Situation have to satisfy. Ratio will be 9:3:3:1. Now, second situation, second place. C active, C active, P does not active. So what is the area I am talking about? I am talking about C active, P is small. Let's say these are the 3 positions in your checkerboard. Let's say this one. These are the 3 areas. Is it came here? C capital and P small. Okay. These are the 3 areas. So what is the character? C active, P is not active. And if P is not active, purple will not form. Situation. So how many in your chart? 3. This is 3. So what is the character? Let's see. C active, P is not active. So C active, P is not active means chromogen will form. Chromogen color is white. So which color will come? White. Clear? Now, these are the 3 locations. 3 locations here. These are the 3 locations. C is small, but P is active. Second position does not matter. Second position only matter if one is recessive. Understand that. If one is recessive, then second position matter. Suppose if I get from my mother side small t, obviously we are curious. Second position. If suppose from my father side, if I get the small t, then my height will be dwarf. But if I get the capital T, then and there my height will be tall. But if the first position, let's say from my mother side I get capital T, bas no matter, enjoy. Whatever will come from mother side, height to tall. Clear or not? Okay. C. So C is not active. If C is not active, then no matter, chromogen will not form, because active C gene will convert the raw material into chromogen. So C not active means chromogen will not form. If chromogen will not form, P cannot active. P cannot work there. Getting my point or not? P cannot work there. P is active, but P only can work upon the chromogen. So there will be no color formation in the flower. So flower without color cannot be transparent. So which color will come? White. So white. Now the last position. Last position. All are recessive. Small c, small c, small p, small p. Already it's given. White. Now already I have written here. White. So what is the final ratio? 9:7 instead of 9:3:3:1. Clear or not? It need to be 9:3:3:1. But here Mendelian law is not applicable at the final round. What we get? 9 purple, 7 white. Okay. So this is based on same dihybrid cross, but this is a situational cross. I have given you one situation. And understand that that type of question might be asked in the exam hall. This is situational cross. If you see in situation homozygous, because only the thing you have to remember, I told you earlier also same thing, alphabet will be changed, A, B, C, D, alphabet will be changed, character will be changed, cross will be remain same, and in standard cross these tricks will be applicable. That's why I have marking with the color that these are the tick position tick mark, minimum 1C and minimum 1P is capital. Clear? Understand or not? This is the 9 area in our chart. So please, these tricks is very much useful. Here, why you will go for cross one question, biology question, we have to solve within 30 seconds. Understand that. Then 90 question can be solved in 45 minutes. Otherwise, if you take, majority of you take 90 question to solve more than 90 minutes. So here, so you will not get the time for physics and chemistry. That is obvious. Traditional process will not be applicable here. Understand that. Every student will go for normal solving, then will try to find out the answer. You have to think something else. Clear? Done? See this chart very carefully. Mendel studied on 7 contrasting characters. So sir, these characters I know. Yes. Every area we have to think which type of question will be asked. Question will not be asked what is the contrasting character of the height, tall and dwarf. That you have to answer. These are not the question. Mendel studied how many color-based experiments. These are the question. No one focus on that part. No one. Whenever we study, we never focus how many color-based experiment was there. Remember that whatever the Mendel discuss here, out of that total from the character, total 7 character Mendel studied. Out of that 7 character, 3 color based. 3 color based. 2 shape oriented. Write down this. 2 shape oriented. No one focus this area. So 3 to 5 done. 1 position of the flower. Position of flower. Next, 1 character, height of the plant. Clear? These are the 7 characters. First character, 3 color based, 2 shape oriented, 1 position of the flower and the height of the plant. Clear? These are the 7 contrasting characters. Now, if we check here, very carefully check this. I told you earlier also Mendel was not biologist. So, whatever the character he given, he have observed this dominant character in a pea plant population. They are high number. That's why they have Mendel discussed that these are the dominant character. So, how we have denote all time? Remember the allele tall given. So, here capital T and here small t. Here violet given dominant, so V, capital V, here small v, it will be not W. Axial, capital A, small a terminal. Inflated I, capital I, constricted small I. Green, capital G, small g, yellow. Round, capital R, wrinkled small r. Yellow, capital Y, green, small y. Out of these 7 characters, understand that the major confusing character is color. 2 color. 2 color means 1 color is pot color, another color is seed color. These are bit confusing. Why? Because you just see pod, this is the pod, okay? Pod color is green, that is which is normally available in the market. Green color pod. Always remember that the green color P, if you have seen the covering, that is the dominant character. Some of them are yellow, but the opposite one, yellow seed is normal, green seed is recessive. So they are opposite in color. Clear? Now check this very carefully. Very important one. Bit quite confusing. Back cross and test cross. So what is discussed in the NCERT? That cross of F1 hybrid back with any of the parent. So who is F1 hybrid? If we just elaborate this part, that back cross stands for F1 hybrid, let's say it's capital T small t, Either it will be cross with capital T or capital T small t. Sorry. Or with the small t small t. Why we do this? Why we do this? What is the necessity of the back cross? To improve varieties. Understand that. To improve variety. Suppose, in a very easiest example I am telling you. I have studied my gene, my family pedigree, and thereby I get to know I am a heterozygous dominant tall. Now I need my child will be tall. Family planning. So obviously I will search for the girl who is homozygous dominant. Getting my point? So by naked eye, I cannot understand. So what I have to do, whatever the girl I will choose, I will first do the pedigree. And if by the pedigree calculation it will come that that girl is a homozygous tall, obviously my offspring will be 100% tall. Getting my point? So by that way, because right now one chapter is deleted from our syllabus, that is improvement of food productions. Okay. So there, these strategies actually applied to get the tall plants. Okay. Any variety if we have to improve, then we will do this kind of homozygous cross. So F1 hybrid with any of the homozygous parent. That is the back cross. But what about the test cross? Test cross is crossing of F1 hybrid with only the recessive parent. Remember that. Only the recessive parent. Now, sir, how? Because here we have to know the unknown genotype. Now, I don't know about my height. I know I am tall, but I don't know I am capital T or capital T small t. So what we will do in that situation for the test cross, that unknown, let's say, that is a probability. If I am capital T, that is well and good. My next generation obviously will get any one of them, capital T. So my next generation also will be capital T. Minimum will carry capital T. Understand that situation. If I am the pure tall, but I don't know. So to cross check it for the, usually this is done for the plant. Okay. Just I am giving you my example. Now I marry with a small t. And the outcoming baby, understand that this is the parental generation. Okay. In the F1 chart, capital T, small t, small t, small t. So capital T, small t, capital T, small t, small t, small t, small t, small t. But if my genotype is capital T, and if I marry with the recessive one, obviously my F1 will be capital T, small t, that is tall. So 100% tall probability is there if I am homozygous tall. But if somehow I am heterozygous, then if I marry with a dwarf female, there is a probability, understand that. Mendel does not discuss that you will have max to max 4 child. 4 offspring means that is a probability Mendel is talking about. So 50% of them can be tall but heterozygous, but 50% probability is there that your child can be dwarf. Mendel is talking about probability. Understand that. So ratio will be 1:1. So thereby, with the help of the test cross, we get to know about the unknown genotype. Unknown genotype always, that I told you, if I ask that one, our cleaner, so you will say, oh sir, it's so small t. But dominating character is always confused, always. So if you have to know which type of dominant, is it homozygous dominant or heterozygous dominant, that time test cross applicable. Because in the test cross, we will use recessive parent in one side. Clear? But in case of back cross, because back cross actually we need to improve the variety. Here please write down, we do it for improvement of the variety. To do it, example of the back cross, to improve the varieties. Purpose of the back cross, to improve varieties. Purpose of the test cross, to know the unknown genotype.